Our next presentation today will be delivered by Dr. Joe Baumert about development and evolution of risk assessment for food allergens. Joe is an assistant professor in the Department of Food Science and Technology and co-director of FARRP at the University of Nebraska. He received his PhD in food science and technology, specializing in food allergy research. He received his bachelor and master's degrees in animal science, specializing in meat science and microbiology. Joe, please go ahead with your presentation.
Thank you, Roland, for the kind introduction, and I thank everyone for attending the webinar today, in which we will begin to talk a little bit about the evolution or development of various risk. So I will build upon what Geert had talked about in looking at various aspects of using threshold data for risk evaluation. So today I'll talk a little bit about, first, an introduction. As we noted early in the days, we wanted to first understand, do food allergens actually follow classical toxicological principles, so that we could apply some of those basic understandings or underpinnings of risk assessment. That then developed into various safety assessment approaches, benchmark dose approaches, and to what currently is believed as the most robust and transparent approach, which is the probabilistic modeling, can be used in conjunction with a benchmark dose approach. With all of these, it does take a number of input variables, one of which Geert had very thoroughly covered on the clinical thresholds aspect, but I'll continue on and look at additional inputs such as the food consumption portion, as well as the concentration and how we estimate how much of that unintended allergen presence may be available. So in the early 1990s and early 2000s, there was very little data on clinical dose response to food allergens. And since that time, we have established a fair amount, or a very nice amount, of clinical data from these low-dose food challenge trials. Again, at that point in time, though, we were uncertain as to whether or not we could apply those toxicological approaches to food allergen risk assessment. Additionally, we were not sure if the common belief at the time, in which we thought that all allergic reactions that could be quite severe would occur at very small exposure doses. And that was more anecdotal data than it was proven in the clinical science. And I'll show you how we've evolved since then to where we're at currently in our thought line. One of the key aspects of the community coming together to talk about risk assessment for food allergens occurred in 2007 in a workshop sponsored by Europroval, ILSI Europe, and the UK Food Standards Authority. This working group came together and had a workshop to really discuss the various approaches that could be applied to food allergen risk assessment. During this workshop, approaches such as a safety assessment approach, which is typically would be a no-L or low-L type approach, in which you are applying very conservatively either the no observed adverse effect level or the lowest observed adverse effect level, and perhaps some uncertainty around that. This is much more of a deterministic type approach, which does not take into account the entire population. Generally, you would use perhaps a single study or a series of studies in which you select likely the most sensitive individual. Again, that doesn't take into account the distribution that we're aware of now or the allergic population in its entirety. So the benchmark dose approach, very similar to the underpinnings that Geert had talked about, in which you can establish a accepted level of risk or a benchmark dose, and also look at establishment of a margin of exposure can be utilized, which the entire distribution or population of that sensitive population can be used. And coupled with that, the probabilistic approach, which I'll talk in more detail about, can utilize the distributions of what we know on the clinical side, as well as what we can use from evaluating the exposure doses. So for input parameters used in this type of risk assessment, of course, we are looking at those clinical threshold studies from low-dose food challenge trials. It's important to note, unlike classical toxicology approaches where you may be evaluating a chemical hazard, oftentimes there we utilize an animal model and extrapolate and use uncertainty factors perhaps to account for that benchmark dose that we're looking at. With food allergy and food allergy risk assessment, the clinical trials are actually done in the human population, specifically the sensitive population or allergic population from that particular group, for example, peanut allergic individuals. So there is not a need to do an extrapolation from an animal model to what we believe it would react in the human models. Important to note that currently there is no good animal model for assessment of human reactivity, so it does not model human allergenicity. So again, we're relying on that sensitive population. Because of that, we believe that uncertainty factors and our conservatism in the approaches that we'll discuss more in detail here in a moment. The exposure assessment, which I'll talk in more detail as well, looks at the food intake, so not only serving sizes, but also the distribution of consumption, because we know individuals will eat on a population basis different portion sizes. Of course, to round out the exposure assessment, we need to know and have an idea of that level of contamination or that unintended allergen presence. So we talk a little bit about how we quantitate that level. Here laid out how the inclusion criteria and establishments of the reference doses took place. And the one thing I wanted to note in this slide is that it was a very thorough curation process with very specific inclusion criteria. We utilized published studies that were in the public domain, as well as unpublished clinical data from various studies that may have been done from a diagnostic series, threshold studies, or perhaps baseline data from some immunotherapy trials. These individuals, of course, had to have inclusion criteria based on history of reactivity to that food or other factors such as serological positive response to the allergen or a skin prick test. We did rely upon the double-blind placebo-controlled food challenge, which has been the gold standard method for challenge for food allergens. But I do acknowledge that we used, in some cases, open or single-blind data for infants or very young children under the age of 3 upon recommendations of clinical allergists in the area. With that, we were able to establish the no-L and low-L values for each individual that went into the dose distribution modeling. But most importantly, of all of this process, we relied upon objective endpoints for each individual. So individuals had to have an objective reaction, something that could be visually observed by the clinician or research nurse and documented, rather than subjective endpoints, something such as an itchy mouth that cannot be measured. And in those cases, we know subjective endpoints can be called differently by various clinicians and nurses. So we know that when we merge data from multiple publications or sources, there can be a great deal of variability. So again, we relied on objective endpoints. In some cases, we relied upon a subjective symptom at the highest dose in the dose regimen that was being used if there was a clear history of objective reactions in the past. So we did utilize statistical dose distribution modeling that allowed us to look at various parametric models for looking at the population distribution overall. And the three common ones we used were log normal, log logistic, and Weibull distributions. We looked at all three models at this point because currently there is no biological reasoning to prefer one model over the other. So with that, we looked at the goodness of fit across all of the models, and especially looked at how the models fit the actual data. So another important point I'll illustrate here shortly. Where the data was available, we looked at both children and adults together, but we also looked at them separately so we could see if there were any major differences. And as Kieran said, in most cases there were not major differences between children versus adults for most allergens. Things like milk and egg, for example, where the populations are primarily young infants and children, we had much more data for those individuals. We didn't have enough to statistically model adult populations, for example. So in this slide, I show the three different distributions. And this point is to illustrate that it is important to look at not only the goodness of fit and statistical parameters that the modeling will give you and the computer statistics provide you, but we have to note that those statistic parameters will give you goodness of fit across the entire distribution. So not only at these low levels, typically where we're most interested in for establishment of a reference dose, but also at the high end of the curve as well. In this case, of course, if we're interested in establishment of a reference dose at, say, the ED1 value, as Hirt had talked about, we see a departure in the green line, this Weibull distribution, which tends to be the most conservative distribution, but it does not model well to the actual data points that we have in our distributions. So in this case, the group would have chose to include or utilize the log normal or log logistic parametric distributions. For exposure assessment, again, our two main components are that food intake. So how much is eaten and how often is it eaten? Because we know that some individuals may not eat a particular product on a daily basis or during each individual's eating occasion. And of course, that level of contamination or of that unintended allergen presence and perhaps frequency can be utilized in these models. So is the contamination in 100% of the product, or do we know that perhaps there was a random distribution of a particular allergen? So if there is a robust sampling plan included, we could include the fact that maybe 25% of the product would have detectable allergen. Of course, accurate exposure assessment is an important component of the overall risk assessment. So we have very thorough evaluation of the clinical data, but we cannot set aside the exposure assessment as it's a vital important part of the overall assessment. So we have to make sure that consumption data is reflective of the population of consumers that were interested for that particular product or product category, and that the consumption data is very carefully calculated and or analytically assessed, and that the right methods are being utilized. So an example for the exposure assessment would be to utilize something such as a consumption database or dietary survey that may be available in various countries. And say there is Danish and Dutch databases, UK databases for consumption, as well as ones here in the US and Canada, for example. These are all based on different dietary surveys that ask several questions on not only what they eat, but we can gather demographic data as well, age, gender, so forth, that can be utilized in the risk model if interested. They all have different setups though, so if we're looking at different databases, we have to be aware that some are 7-day diary records, others are 2-day, some are a 24-hour recall database. The primary goal, again, is to look at what we know about consumption from a population standpoint for what we believe would be similar to our allergic individuals. So here Ted provided a very nice illustration of why it's important with his example in bread. We rely upon a recommended serving of bread, which would be 1 slice in many countries. We know that individuals may eat 1 slice, but most individuals eat 2, 3, 4 slices, for example. So we need to take that into account into a thorough risk assessment. For the food intake data, what we're looking at, again, is data from the general population. There is some data available on individuals that may indicate that they have an allergy, but that's not the primary goal of most of these consumption surveys. It's more of a nutritional survey. So it's generally unknown that whether or not an individual is truly allergic in that entry. So from this point, we assume that the food allergic individual that would choose to eat a certain product, so if they go out to the market and buy that product, they would consume that same portion size within the same frequency as the non-allergic people that may be in the majority of that general population in the database. So there are efforts to begin to populate and get a better understanding about what eating and consumption habits are for allergic individuals, but we don't have that data currently. For the second part of exposure assessment, of course, the contamination data becomes quite important. So we have to have a thorough understanding of the concentration of that food residue. Specifically, what we're interested in is the protein from that allergenic source to couple with the values that we have from doses on our threshold standpoint. So those are milligram doses of protein from that allergenic source. So we can determine this, the concentration either by calculation, if we know that there was a formulation change or a product changeover, and we can calculate how much may be in the next product after changeover, or we can do quantitative analysis utilizing various analytical methods that I'll discuss in more detail. If using an analytical method, it's important to understand the differences in those type of analytical methods, what they're actually detecting and what they're actually reporting. It becomes very important to make sure that we're coupling a part per million level from protein from the allergenic source in conjunction with grams of product eaten to figure out the exposure dose in a milligram value of protein from that allergenic source. If we don't make the proper conversions, there can be several fold difference in the analytical result that you have and calculation of your exposure dose. So it's important to understand, is the analytical method looking at total protein? Is it looking at detection of a specific protein fraction from that allergenic component, such as casein, for example, but reporting units in perhaps parts per million of non-fat dry melt? So the analytical component can be very challenging to interpret, and I pose caution in ensuring that you're actually looking and doing diligence to understand what it's detecting. So finally, when we get to taking that threshold data and doing our distribution modeling, there can be two different approaches taken, as mentioned earlier, on evaluation of risk. It can be the method one, in which we evaluate and have a reference value for a benchmark dose, say the EDO1 values that are our accepted level of risk, perhaps, and then look at more of a deterministic standpoint. So is the reference dose in comparison to what we know about the exposure dose? So do we have perhaps a risk in that regard? We can use more intricate models, such as the probabilistic model that takes into account all of the distributions of consumption, the concentrations or part per million levels of the unintended allergen, as well as the threshold data, which gives us a much more thorough and robust analysis. So,
Joe, we are approaching the end of your allocated time.
Yep, and this is my last slide. So I provide a picture or illustration of probabilistic risk assessment that was developed by T&O. And this illustration gives a nice pictorial view of what's going on in a probabilistic model, which essentially, if you're using Monte Carlo analysis to look at a random sampling of a threshold value in conjunction with what we know about the allergen intake or exposure dose, which again uses the distributions of the dietary surveys or consumption, as well as what we know about the analytical assessment of that unintended allergen presence. We can have a very robust analysis using all of those distributions and what we know about differences in the allergic individuals as well as their consumption values for the risk assessment. And this was utilized as an underpinning in a lot of the work that was utilized by the VITAL group to establish those reference dose values and validate the consumption. So again, we have very good fits for 11 different allergens in my view, with a couple additional ones that we have some data and we'll continue to build upon that clinical data. But again, we're using the human population, so we are very conservative, feel that there's no need for additional uncertainty factors in this regard. And these reference doses coupled with probabilistic modeling provide a realistic basis for determining reference doses, as well as how we could utilize the data for evaluation of when a precautionary statement should be utilized and provide much more transparency in that endpoint that can be used operationally by the food industry. So as with here, I'd like to acknowledge a number of our collaborators, specifically T&O, Unilever, the VITAL, Scientific Expert Panel and VITAL Group, and ILSI Europe,